Hello everyone. Today we will discuss about sodium. So body water is found mainly in intracellular and extracellular compartments. In intracellular and extracellular compartments. A small amount is found in interstitial fluid compartment. So, body water is found mainly in intracellular and extracellular compartments. A small amount is found in interstitial fluid compartment. This sodium is the chief electrolyte found in large concentration in extracellular fluid compartment. Sodium is the chief electrolyte found in large concentration in extracellular fluid compartment. Coming to the sources of sodium. Table salt is the major source. This sodium is widely distributed in food material. It is more in animal sources. More in animal sources. It is also found in cheese, butter and cocoa. Coming to daily requirement. For adults, it is about 1 to 3.5 gram. For children, it is 0.3 to 2.5. And for infants, 0.1 to 0.5 gram. This is the daily requirement of sodium. Coming to the absorption of sodium. Sodium is absorbed by sodium pump. It is absorbed by sodium pump. This sodium pump is situated in basal and lateral plasma membrane of intestinal and renal cells. This sodium pump is situated in basal and lateral plasma membrane. Basal and lateral plasma membrane of intestinal and renal cells. This sodium pump actively transports sodium into the extracellular fluid. Sodium pump actively transports sodium into the extracellular fluid. So let's see the sodium pump. So, this sodium pump is also called as sodium potassium ATPase. It is also called as sodium potassium ATPase. It requires ATP and Mg++. ATP and Mg++. This intracellular sodium concentration is around 10 millimole. Intracellular sodium concentration is around 10 millimole. Extracellular sodium concentration is 150 millimole. There is high concentration of proteins and phosphate anions inside the cell than outside. Concentration of potassium inside the cell is 100 millimole inside the cell and the outside is 5 millimole. Thus, the sodium pump maintain both magnitude and direction of transmembrane concentration gradient of these ions, that is sodium and potassium. Then coming to the sodium pump is an enzyme, that is sodium potassium ATPase. It is a glycoprotein. It is a glycoprotein and it composed of 2 alpha and 2 beta chains. 2 alpha and 2 beta chains. Its activity depends on the presence of this sodium and potassium. Its activity depends on the presence of sodium and potassium. It requires ATP and Mg++. Mg++ as cofactor. This enzyme, that is sodium potassium ATPase, it hydrolyzes a high energy phosphate bond of ATP. It hydrolyzes a high energy phosphate bond of ATP and the released energy is used for transporting 3 sodium ions outside and simultaneously 2 potassium ions inside across the cell membrane. So this enzyme sodium potassium ATPase, it hydrolyzes a high energy phosphate bond of ATP and the released energy is used for transporting 3 sodium ions outside and simultaneously 2 potassium ions inside across the cell membrane. In this way, each sodium potassium pump transfers 9,000 sodium ions outside and 6,000 potassium ions inside the cell in 1 minute. Each sodium potassium pump transfers 9,000 sodium ions outside and 6,000 potassium ions inside the cell in 1 minute. This sodium pump is very active in cells. Sodium pump is very active in cells where activities depend largely on transmembrane sodium influxes. Transmembrane sodium influxes. So this sodium pump, it is active, very active in cells where these activities depend largely on transmembrane sodium influxes. For example, muscle fibers, renal tubule cells and intestinal mucosal cells. So let's see the forms of sodium pump and mechanism. Mechanism of action of this sodium pump. This sodium potassium ATPase exists in 2 forms, that is E1 and E2. Sodium potassium ATPase exists in 2 forms, that is E1 and E2. First we will see the E1 form. E1 form. Its ion binding and phosphate binding sites present on the cytoplasmic surface of the membrane. The E1 form, its ion binding and phosphate binding sites present on the cytoplasmic surface of the membrane. 3 sodium ions from the cytoplasm. 3 sodium ions from the cytoplasm bind with the ion binding sites of E1. Bind with ion binding sites of E1. 3 sodium ions from the cytoplasm binds with the ion binding sites of E1. This leads to phosphorylation of aspartate residue of E1. Phosphorylation of aspartate residue of E1. So by the binding of this sodium, 3 sodium ions, it leads to phosphorylation of aspartate residue of E1 with the help of this ATP and Mg++. This phosphorylation of the aspartate residue of E1 results in conformational change in E1 and E1 is changed to E2 and it forms E2. So now E2 exposes both ion binding site and phosphate binding sites on the extracellular surface of the membrane. So this E2 exposes both ion binding and phosphate binding sites on the extracellular surface of the membrane. It lowers the affinity of ATPase for sodium. It lowers the affinity of this ATPase for sodium, and it releases sodium into the extracellular fluid. So thus this sodium enters into extracellular fluid. Now the potassium ions. The potassium ions from the extracellular fluid bind to the respective ion binding sites of the pump. These potassium ions bind to the respective ion binding sites of pump. This lowers the affinity of E2 for phosphate, and this phosphate is released into the extracellular fluid. This dephosphorylation here, so dephosphorylation. So this dephosphorylation changes E2 into E1, and this lowers the affinity for K plus ions. So K plus ions released into the cell, into the cytoplasm. So this leads to release of the K plus ions from ATPase into the cell. That is the mechanism of action of this sodium pump. Then inhibitors are there for the sodium pump. First one is ORBane. ORBane. ORBane is a cardiac glycoside. It is a cardiac glycoside. This inhibits the sodium potassium pump by blocking the step of dephosphorylation. This inhibits the sodium potassium pump by inhibiting the step of this dephosphorylation. Next inhibitor is vanadate. This inhibits the pump when present inside the cell. When present inside the cell, this vanadate inhibits the sodium potassium pump. Then next we will see excretion of sodium. Coming to excretion, every 24 hours approximately 25,000 millimole of sodium are filtered by the kidneys. Due to tubular reabsorption, less than 1% of this sodium appears in urine. That is about 100 to 200 millimole per day. So every 24 hours approximately 25,000 millimole sodium are filtered by the kidneys. Due to tubular reabsorption, less than 1% of this sodium appears in urine. That is about 100 to 200 millimole per day. Approximately 70% of the filtered sodium is reabsorbed in proximal tubule. Proximal tubule. 70% of the filtered sodium is reabsorbed in proximal tubule. About 20 to 30% of the filtered sodium, it is reabsorbed by ascending loop of Henle. Ascending loop of Henle. So that is the excretion. Coming to the functions of sodium. First function is fluid balance. Fluid balance. Sodium maintains crystalloid osmotic pressure of extracellular fluids and it helps in retaining water in extracellular fluid. It maintains crystalloid osmotic pressure of extracellular fluid and it helps in retaining water in extracellular fluid. Second function is neuromuscular excitability. Neuromuscular excitability. Along with other cations such as potassium, calcium, magnesium and H+, this sodium is involved in neuromuscular irritability. Next function is maintenance of viscosity of blood. Maintenance of viscosity of blood. The salts of sodium with globulins are soluble. Then further, sodium and potassium both regulate in maintaining the degree of hydration of the plasma protein. That is a function in maintenance of viscosity of blood. Next function is role in resting membrane potential. Resting membrane potential. This plasma membrane has a poor sodium permeability and passive sodium inflow through it. Plasma membrane has a poor sodium permeability and passive sodium inflow through it. This sodium pump keeps sodium concentration higher outside than inside. This separation of charges is called as polarization of the membrane. Sodium pump keeps sodium concentration higher outside than inside. This separation of charges is called as polarization of the membrane. It creates a potential difference of 70 to 95 millivolts across the membrane. This creates a potential difference of 70 to 95 millivolts across the membrane. This is called as resting membrane potential. The next function is role in action potential. Role in action potential. A local depolarization of nerve fiber or muscle fiber is seen in stimulation. A local depolarization of nerve fiber or muscle fiber, it is seen in stimulation. This rapidly increases its permeability to sodium and it causes considerable transmembrane influx of sodium down its inward concentration gradient. That is the role in action potential. These are the functions of sodium. Coming to clinical aspects, there are two types of clinical conditions, hypernatremia and hyponatremia. Hypernatremia and hyponatremia. First we will see hypernatremia. This is high sodium concentration. High sodium concentration. It does not mean that the total body sodium content is increased. It means extracellular sodium is excessive relative to water. It does not mean the total body sodium is increased. But it means that extracellular sodium is excessive relative to water. So decrease in body water and increase in body sodium. Decrease in body water and increase in sodium. That is hypernatremia. So let's see the conditions where this hypernatremia occurs. First condition is simple dehydration. Simple dehydration. This occurs as a result of excessive sweating. Excessive sweating with inadequate water replacement. Excessive sweating inadequate water replacement. That is simple dehydration. Next one is diabetes insipidus. This is characterized by lack of ADH, that is antidiuretic hormone, or failure of the hormone to act on the target cells. It occurs usually as a complication of pituitary surgery. As a consequence of pituitary surgery when hormone is not produced in adequate amount. In nephrogenic diabetes insipidus, the kidney cells are unable to respond to the hormone. That is in diabetes insipidus. Next one is osmotic loading. Osmotic loading. If the kidney excrete large quantities of very soluble substances such as glucose, urea, amino acids, then osmotic effect of these substances on the urine causes co-excretion of large amounts of water. If the kidney excrete large quantities of very soluble substances, for example glucose, urea, amino acids, then osmotic effect of these substances on the urine causes the co-excretion of large amounts of water. Relatively little sodium is excreted, so the plasma level rises. In case of very ill patients on high protein diet, urea is formed, which is then excreted in very large amounts along with large volumes of water. That is osmotic loading. Next one is excess sodium intake. Excess sodium intake. Normally in clinical medicine, 0.9% of sodium chloride it is administered intravenously. That is 154 milliequivalents per liter. 154 milliequivalents per liter. So normally in clinical practice, this 0.9% sodium chloride is administered intravenously. That is about 154 milliequivalents per liter. This excessive use of isotonic saline, particularly in children, it leads to hypernatremia. This hypernatremia can also occur following administration of sodium bicarbonate. Sodium bicarbonate in acidosis treatment. So that is the excess sodium intake. Next one is steroid therapy. Steroid therapy. Mineralocorticoids control the metabolism of sodium. These mineralocorticoids also cause the kidney to absorb sodium from the glomerular filtrate. It results in increased plasma concentration. Mineralocorticoids cause the kidney to absorb sodium from the glomerular filtrate. So it results in increased plasma concentration. In certain tumors of this adrenal gland, large amounts of aldosterone is produced. It is called Cone syndrome. Certain tumors of this adrenal gland, large amounts of aldosterone is produced. That is called Cone syndrome. That is about hypernatremia. Next we will see hyponatremia. Hyponatremia. This means a low plasma sodium concentration. So let's see the conditions where hyponatremia occurs. First one is diuretic medication. Diuretic medication. One of the most common causes of hyponatremia is the use of diuretics. Many diuretic medications act by promoting sodium excretion by the kidney. So these diuretics that acts by promoting sodium excretion by the kidney. Here total body sodium will be reduced. But as the extracellular volume reaches critical dimension, there is a counter effort to retain water which then dilutes the sodium, and here hyponatremia occurs. Next one is excessive sweating. Here loss of fluids of this high sodium and potassium, sodium and chloride content like sweating, but are replaced by salt deficient fluids such as water by mouth or glucose solution by IV. Loss of fluids of high sodium and chloride content. Then these are replaced by salt deficient fluid such as water by mouth or glucose by IV. This causes hyponatremia. Next one is kidney diseases. kidney diseases. In glomerulus, blood is filtered and sodium is reabsorbed by the renal tubules. In glomerulus, blood is filtered and sodium is reabsorbed by the renal tubules. Due to kidney dysfunction, sodium is not reabsorbed and it is excreted in the urine. There is also failure to excrete water. So this also causes hyponatremia. Next one is congestive cardiac failure. This hyponatremia is common in heart failure. Two reasons are there. First one is diuretic administration. Then in advanced stages, congestive heart failure causes low sodium concentration. It is because the low cardiac output is sensed incorrectly by the brain as low blood volume. And there will be increased secretion of this ADH. So this all results in hyponatremia. Next one is gastrointestinal loss. Gastrointestinal loss. Prolonged and severe diarrhea results in reduced sodium or chloride levels in plasma and extracellular fluid. This causes hyponatremia. So these are all about sodium. This is today's topic. Thank you for watching.